let's be honest, we've all had an episode of diarrhea at least once in our lifetime. But can you imagine having explosive, non-stopping diarrhea for weeks on end with no relief? That's what it's like for people who have an antibiotic-associated disease called Clostridium difficile infection, or CDI. There are over 500,000 cases of CDI reported per year and over 29,000 deaths per year, which can add up to $3.2 billion in annual cost burden to the US healthcare system and ultimately to you. On September 21st, 2016, the United Nations declared that antibiotic resistance is the greatest and most urgent global risk. And CDI happens to be a disease linked with both antibiotic use and resistance. CDI is caused by rod-shaped bacteria called Clostridium difficile, or more commonly known as C. diff. A key characteristic of C. diff is that it is able to form these tough and round structures called spores that are the infectious form of the disease. Spores are basically like super eggs that can survive in many different environments until a trigger causes them to hatch, or what we call germinate, into toxin-producing cells that can eat away at the intestines. This germination process is required for CDI symptoms to occur, and it just so happens that the gut can be a perfect place for these spores to germinate in. My research focuses on how bile salts that are naturally produced in the liver and secreted into the intestines have potential in the preventative treatment of CDI. In our bodies, there are bile cells that act as natural germinators of C. diff spores and bile cells that act as natural inhibitors of spore germination. In healthy individuals, there are good bacteria in the gut that help balance these bile salts so that the spores do not end up germinating in the gut. However, in patients who are immunocompromised, like those who have recently taken antibiotics, that natural gut bacteria is now depleted and is much less efficient at regulating these bile salts, so the spores end up germinating into those toxin-producing cells that cause CDI symptoms. Our lab aims at producing synthetic bile salt compounds that can act as better germination inhibitors than the natural inhibitor. One compound, called CANSA, work very well with one strain of C. diff, but not as well with other strains. Currently, we're testing a new compound called 07C that has been able to prevent spore germination and CDI symptoms in mice with various strains of C. diff. We hope that by continuing to test new bile salt compounds, that we will be able to find a potential solution to this very poopy problem. Thank you.